biological molecules unit. This video 1.10 I believe it is, which is about nucleic acids, which is going to cover both DNA and RNA and their structures and their role in the cell. So to start off then, you should hopefully already know that the DNA structure is this double helix structure from GCSE. And if you look at the bottom down here, you can see that it's made up of individual bits called nucleotides. So this bit I'm just circling now is one nucleotide made up of a phosphate group, a pentose or a 5-carbon sugar, and an organic base. And they make the two strands, which then spiral up together to make what we call a double helix. And this structure was worked out, I suppose, and calculated first of all by Watson and Crick in 1953. They didn't do it alone, however. They also did it with two other scientists. Those scientists were was Maurice Wilkins and Rosalind Franklin, and Rosalind Franklin paid the ultimate price because she worked so much with X-rays that she actually developed cancer as a result, and therefore got a Nobel Prize posthumously for her work on this. So today we're going to look at DNA and then RNA. So the function of DNA effectively is that it stores your genetic information. So it's a long chained molecule storing all our genetic information, which allows us to grow and develop and controls everything our cells do. So if we're looking at nucleic acid, a nucleic acid is the polymer, and a nucleic acid is a long repeating unit. So DNA and RNA are nucleic acids made up of monomers called nucleotides. Okay, and effectively one nucleotide, if we have a look at this picture, is made up of the following things. So one nucleotide is an organic base, and I would always call it a nitrogen containing organic base or a nitrogenous organic base. So if you're asked the question, describe the structure of a nucleotide, I'll say it's a nitrogenous organic base, and you could name the bases, which we'll talk about in the next few minutes. It's made up of a 5-carbon or a pentose sugar, and that's deoxyribose for DNA and ribose for RNA, and it's made up of a phosphate. Now you won't be expected to draw it to the detail of the other pictures, but you would be expected to describe it. So the answer for what one nucleotide would be is here in the blue. So we need to know obviously that the nucleic acids are two forms, DNA and RNA. And almost all of DNA in eukaryotic cells is found in the nucleus, but there is some in the chloroplast of plant cells and the mitochondria of animal cells. And there are 3 types of RNA, which we'll talk about later on. Just to remind you then that DNA and RNA contains a 5-carbon or a pentose sugar called ribose or deoxyribose. And then if we look at the structures of the nucleotides, they're very, very similar. So you can see both DNA and RNA. If we look at DNA first of all, so if we look at these ones first of all, DNA, all of the nucleotides, there's 4 different types have a phosphate. They all have a deoxyribose sugar, so they're identical apart from the bases. So we've got 4 different bases, sorry. So we've got adenine at the top, we've then got cytosine, the C below, and then the G for guanine and the T for thymine. And if we look at RNA, you'll see that the structure is very, very similar, but obviously there are subtle differences which you would be expected to discuss. So they both, just like DNA, they have a phosphate group, except for the sugar this time is the ribose sugar in the middle, and then the bases are the same, but if you have a look at the bases, they have an adenine base, they have a nucleotide of that, they have a cytosine base, they have a guanine base, but then instead of thymine, they have uracil, which is a very similar molecule, but very, very slightly different in its atoms. So there's only 4 real different types for RNA and DNA. The phosphate's always the same. There's always a pentose sugar, which is either deoxyribose in DNA or ribose in RNA. And there are 5 different bases. As I said, uracil is in RNA, whereas thymine is in DNA. Adenine, guanine, and cytosine is the same in both DNA and RNA. So when we make the nucleic acid then, so just a reminder in the top right hand corner, we've got this double helix structure. And if you look at the double helix structure, it's the same on each side. So if we look at just like this side here for the moment, this is basically what it's made of. So you can see here, if I draw a line here, you've got a nucleotide there, you've got a nucleotide there. So how do we join the nucleotides up? Well, it's just like joining up molecules of sugar, monomers to make disaccharides, monosaccharides to make disaccharides, sorry, or amino acids to make dipeptides. It again is the removal of water in a condensation reaction. And this forms what we call a phosphodiester bond. So we call it often the sugar phosphate or the phosphodiester backbone that makes up the DNA strands. And basically, a phosphodiester bond is slightly more confusing because it's one phosphate molecule with a covalent bond either side, and the covalent bond is an ester bond. So one phosphodiester bond is phosphate with two ester bonds either side. And then you've got in the middle your bases, which obviously are the letters, and that's the coding. And obviously then you'd have hydrogen bonding to combine the bases with their complementary base pairs. So let's have a look at the organic bases then. So basically there are two groups of bases which you need to know. So you've got purines, okay, which are in the blue box below. So the purines, you can see they've got adenine and guanine. So the adenine and guanine have what I would say is a double ring structure of carbon. So their purines have got the bigger structure, effectively. Purines have got a double ring structure, so that's both the same in adenine, and then there's a double ring structure in guanine as well. So A and G are purines, and they have a double ring structure. The pyrimidines, which are in the red box below, they are T, C, and U, obviously because T and U replace each other. So remember that T is only found in DNA, whereas U is found in RNA, and I'll describe them as single ring structure. Again, you don't need to describe the structure. You just need to be able to describe the whether they're single or double ring structures, whether they're purines or pyrimidines. If you just look at the difference, it's a good time to show the difference between thymine and uracil there. So there's just one area different. They have some carbons and an extra hydrogen or two in there, but you don't need to know that. You need to know that they just replace each other. So why is this important? Why is it important that we have purines and pyrimidines? Well, effectively, it's because the DNA strand is effectively like a ladder, okay? And having the purines and pyrimidines is quite vital. If you look at this picture before we get the writing up, you can see that the complementary pairing always involves a purine and a pyrimidine. So we've got a purine here in G, which is a double carbon structure. So if I draw that as like that, matching up with a single carbon pyrimidine in C. And then below it, we've got a double carbon A, which is a purine again, combining with a single carbon T. And then below that, just the opposite way around, you've got a single carbon pyrimidine T binding with a double molecule of adenine, which is a purine. Now that's really important because what that does is it means the width of the ladder doesn't change. So you've always got 3 carbons of organic bases between them, and obviously if it changed, that would cause the ladder to sort of wiggle, and that wouldn't make it a very stable structure. You wouldn't want to climb a ladder that doesn't have straight rungs. Now also between the carbon, the nucleotides then, are hydrogen bonding. So between G and C, you can see in this diagram, there are 3 hydrogen bonds every time. Between those complementary base pairs, and between A and T there's always 2 hydrogen bonds. And again, individually hydrogen bonds are very weak, but when you've got millions of them between all the length of base pairs, then you will have a very strong stable structure as a result. You can also see the phosphate sugar backbone goes upwards on this one and then downwards on the other side, so we call it an anti-parallel structure. Okay, so how is it adapted for storage? Well, it's got 2 nucleotide strands that come together like a ladder. The phosphate sugar backbone makes the uprights. The bases are like the rungs of a ladder. Hydrogen bonds form between the bases, making the structure very stable due to their high number, but it also allows it to be unzipped when we talk about DNA replication next time. And you've got strong phosphodiester covalent bonds between the sugar and the phosphate groups, which are those ester bonds. And then it's vital because it obviously needs to be stable to be the structure of an organism. If it was unstable, it would cause copying errors, which we would call mutations, which, as we know, can lead to cancer if you get mutated DNA. So we've talked about this already. So the hydrogen bonding is really important. You learn that A to T is 2, G to C is 3. So A to T 2, G to C 3. And that obviously again just allows that strength, and it's always a purine and a pyrimidine, which always keeps the width of the DNA molecule the same. So A to C2, G to C3. And that's what we call the complementary base pairing. So anytime you describe the structure of DNA or anything to do with DNA, I would always talk about complementary base pairing in there, because it's normally a bonus mark, even if it's not too obvious. So structured DNA then is our information code, and effectively it's a really long molecule, which means it can store loads of information. The base pair rule means that we can replicate it, which we'll talk about next time, and it's a double helix structure, which gives the molecule lots of stability. And hydrogen bonds allow easy unzipping, which we'll talk about next time as well. So there's a task for you to do if you want to do that. You can pause it and have a look at that, but otherwise I'm just going to keep talking now about RNA. So there are 3 forms of RNA. RNA is messenger RNA, ribosomal RNA, and transfer RNA. You'll learn about transfer RNA much more in year 13, where it brings in complementary amino acids to make the proteins in a process called translation. Ribosomal RNA basically makes your ribosomes, but what we're interested in more at the moment is messenger RNA. So how is RNA, what's the structure like? Well, it's very similar to DNA. We already know that RNA has a ribose sugar rather than a deoxyribose, but still a 5-carbon sugar. The main role of, in this case, messenger RNA is to transfer genetic information to the ribosomes in protein synthesis. And the key thing you need to know for RNA's job of coding for proteins is that every 3 RNA nucleotides, every 3 bases, organic base in the RNA, which is called a triplet or a codon in year 13, codes for 1 amino acid. So this is just a really brief introduction of it, just so you know. You'll do this much more next year, but effectively what you need to know is that the gene, which is 1 characteristic's worth of DNA, is copied to make messenger RNA in a process called transcription. That happens in the nucleus, and the reason you only copy 1 gene's worth of DNA is because the DNA is too big to leave the nucleus. That messenger RNA then goes to the ribosome, where it is used, and that code of organic bases on the messenger RNA is used to put the amino acids into the correct order. But you'll do that much more later. But the key thing is just knowing that every 3 bases of the messenger RNA codes for 1 amino acid. And this is just again to show you that. So these are the 20 amino acids you don't need to know about. And effectively, let's say you had wanted to make valine. Okay, this just shows you that you could have the following code, let's say G, U, and C, and that would code for valine. Okay, but you could also do G, U, and A, and that would code for valine. That's what we call a degenerative code, which means that it can have more than 1 way of making coding for the same amino acid, and that code is the same in all living organisms. So how does messenger RNA and DNA differ? Well, DNA we've said is a double-stranded molecule, whereas RNA is only single-stranded. DNA is long, so DNA, effectively each chromosome is a really long chain of billions of nucleotides long, whereas RNA is short because it's only coding for 1 gene, 1 characteristic alone. So the smallest one that I'm aware of, it would be the gene for insulin, for example, which is about 50 amino acids, about 150 150 nucleotides of RNA stuck together. It's got thymine in DNA, whereas RNA's got uracil. The 5-carbon sugar is deoxyribose in DNA, whereas it's ribose in RNA. It is found in the nucleus only, pretty much, the DNA. We did say also the chloroplast and the mitochondria, mainly in the nucleus, whereas RNA is found around the cell because it has to move to the ribosomes to cause protein synthesis. DNA is a stable and permanent structure, obviously really important, and it can be replicated and it has to be stable to do that, whereas RNA is not stable and it's a temporary structure because it's only used until the protein's made and then it's broken down and recycled. There's only 1 type of DNA, where there's 3 types of RNA. And the last sort of thing is, imagine you've got this question: how does DNA in eukaryotes and prokaryotes differ? So the reason I want to show you this is just to remind you of the knowledge you already have about this. So in eukaryotes there are lots of genes, so it'd be a long structure. DNA is mainly in the nucleus in the eukaryotic cell. It's got lots of chromosomes, which is a member of DNA associated with the histone protein, which is the chromosomes, and it contains non-coding introns as opposed to exons, which you'll do about later on. There are very shorter shorter strands of DNA in prokaryotic cells because there are fewer genes. The DNA is loose in the cytoplasm. The DNA is naked, which means that it's not associated with histone protein, and it is also circular DNA, so you have plasmid molecules as opposed to linear DNA structure and chromosomes in eukaryotic cells. And it does not have non-coding regions. So just to finish that bit off, just to explain a bit more, you will do that in the future. But DNA, there are coding bits of DNA called introns, coding bits of DNA called exons, which basically create our amino acid, put our amino acids together to make proteins. And then there are non-coding introns, which we will discuss more later on in A level, but do other things other than make proteins. So that's been useful, and I will see you next time looking at how DNA replicates.